Hey, all, Connor Wander here with the Straight from a Scientist podcast. It's episode 55. It's with Dr. Scott Parnell from the Bowles Center for Alcohol Studies right here at UNC. I sat down with Dr. Parnell to talk about his work on cannabis and alcohol and how those two can both work to really harm embryos and fetal development. So as babies develop in the womb, they're really, really sensitive to a lot of substances like drugs, and cannabinoids like those found in marijuana, CBD and THC are becoming increasingly more popular, and we need to really know what they're doing to fetal development. And preliminary studies don't look so great in that it seems like cannabinoids are working in the same way that alcohol does. We've known for a very long time that alcohol causes severe damage to a developing embryo and can cause a lot of birth defects and associated with fetal alcohol spectrum disorders. And so yeah, it was a really, really interesting talk, and I think you're going to really enjoy it, and it's kind of eye-opening for me as well. With that, I'll give it over to the podcast. Again, you can find us on iTunes, YouTube, Spotify, Stitcher. Let us know if there's any place you want to see us on, and enjoy. Welcome, everyone. Straight from a Scientist podcast here. I'm Connor Wander, here sitting with Dr. Scott Parnell from the Bowles Center for Alcohol Studies. We're in his office, and we're going to talk about fetal alcohol syndrome, which is and the greater spectrum of disorders associated with alcohol use during pregnancy, as well as the implications of marijuana use and the compounds like THC and cannabidiol within marijuana that could also be causing issues embryonic development. Thank you, Dr. Parnell, for coming on. This stuff is really important.
Happy to do this.
So, could you explain fetal alcohol syndrome and kind of some of the issues with drug use, embryonic development?
Right. So, fetal alcohol syndrome was first diagnosed a little more than 40 years ago, and it was identified due to its very characteristic craniofacial birth defects, along with low IQ, learning and memory problems, etc., as well as delayed growth problems. But over the years, we've realized that fetal alcohol syndrome, as defined by particularly the craniofacial birth defects, is really just one small part of the larger problem. And we've learned that there are many, many children who are affected by prenatal alcohol exposure who don't have the craniofacial birth defects that we see in fetal alcohol syndrome. And so we've come to identify a lot of these kids as having fetal alcohol spectrum disorders, which is the wider spectrum of what alcohol does. And we've learned over the years that the craniofacial birth defects that partially define fetal alcohol syndrome only occur when the embryo is exposed to alcohol during the 3rd to 4th week of early development. But what we've known, or what we've learned over the years, is that the developing brain and many of the other parts of the body are sensitive to alcohol exposure at any point during development. So that's why we've had to reclassify fetal alcohol syndrome as being just one aspect under the greater fetal alcohol spectrum disorder umbrella. Simply because while the window to affect the face is very early, the brain as a whole can be affected at any point in development.
Sure. So yeah, alcohol is supposed to basically affect every tissue in the body, right? So the surface levels, sounds like that's only during first trimester.
But that's particularly true of the face. During very early embryonic development, the face and the brain are developing together, and they're undergoing some very critical developmental processes that, if disrupted, cause the rather severe brain defects and craniofacial defects that we know as fetal alcohol syndrome. But we also now know that the brain can be affected at any time.
And even after development, just a small segue, some labs next door to us study intermittent ethanol binge drinking in adolescence. And so the brain is sensitive all the way down, but perhaps a little more sensitive during those early embryonic development times.
Right. While early embryogenesis is certainly the most critical window for some of the really severe brain defects that we see in fetal alcohol syndrome, we know that there's no safe time for alcohol consumption, and that includes after birth too. That's why we don't let children drink. We don't let children drink, at least.
Mm-hmm. We've known about FAS and FASD for I think around 40 plus years.
Right, since the early 70s.
Early 70s, and so before then, people just didn't know the dangers. That was around the time we discovered the dangers of cigarettes, right? So
it was. It was about the same time that people cautioned women to stop smoking when they found out they were pregnant. And we now do the same thing for fetal alcohol syndrome. And there's actually a lot of good evidence that people who are drinking a lot tend to stop drinking, or at least slow down their drinking once they find out they're pregnant, which is very good, because that means that you limit the effects of alcohol. Unfortunately, some of the severe craniofacial and brain defects that we might see with prenatal alcohol exposure may occur before some people realize that they're pregnant. So it's important to, if you're thinking about getting pregnant, to slow or stop drinking while you're trying to get pregnant.
Yeah, and unfortunately, it's so hard to just predict some of these things. I think to rattle off a stat that may not be accurate, I can fact check that a good portion of pregnancies are unplanned. So these pregnancies are essentially, no one can prepare their body and their habits.
Absolutely, and that makes our job a little more difficult, because I think the CDC states that about 50% of pregnancies in the U.S. are unplanned. And so to me, that means that we need to be careful, particularly if we're not on birth control or not using birth control, to limit drinking as best we can. It's not always possible, but and things happen. But having said that, I still think it's important that if you do find out you're pregnant, to stop drinking.
Yeah, I think you touched on that CDC. The official CDC recommendation, as far as I know, is that all women of childbearing age and not on birth control don't drink at all, which isn't exactly realistic. Yeah, it's optimally, maybe that's where we would be at, but that's a far tale to, or like a tall order is ask of a greater populace.
It is, and it's probably not feasible. I think that as a whole, if possible, to use birth control, and I think that the use of birth control is not just for women. Men can also use birth control, and I think that's important for men to take responsibility there as well.
Yeah, and we often think of it as purely a female thing, but obviously takes two to tango. And
right.
Without getting too preachy, I can't help but think of the old Spider-Man quote that's now a meme: with great power becomes great responsibility. And that's on males and females, I think, as well. These things, because they can happen so early and before actually people detect them, I just hope that people use caution. That would be my my perspective here. I want to lecture.
It is, and this is not just about. It's not just true for alcohol either. Right. Many things can affect early embryonic development, classic case is Accutane, which was used for acne,
Right.
and still is used for acne. But we found out pretty quickly that Accutane causes birth defects very similar to what alcohol does. So it's important to limit what we put in our bodies, particularly if we're thinking about getting pregnant or if we find out you're pregnant.
Excuse me. So I know a lot of the work that you study or the work that you do is studying how alcohol and these other drugs affect the central nervous system during development. Perhaps you're thinking there's a way that we can intervene in the process?
Maybe. A lot of the work that my lab does is studying how alcohol and other drugs impact early development on a cellular and molecular level, with the hopes of preventing the effects of these drugs, or at least reversing them if possible. We also study genes that may play a role in prenatal alcohol exposure. We know that there is a large genetic component to fetal alcohol syndrome. Not everyone who is equally exposed develops FAS. Some of that is how much we drink, and we know that more alcohol is worse than a little bit of alcohol. That shouldn't come as a surprise. We know that there are nutritional factors that play a role, but we also know that there is a large genetic component to fetal alcohol syndrome, and that some people will not develop FAS as easily as others.
So some are more vulnerable genetically, and it's hereditary. So.
To some extent, yes, and obviously alcohol or other drugs are the key factor there, but we know that there's genetic components, and my lab is trying to figure out what those genetic components are, with the ideal of hoping that they can lead us into mechanisms that can help prevent fetal alcohol.
Sure. Yeah, because maybe you can find genes that confer resistance. So embryos that, even when exposed to alcohol, again, I'm just kind of spitballing here, would not have as many birth defects down the road. I think, especially since it is so hard to kind of know, especially in early embryonic development, what's going on if you're pregnant at all. By the time some parents find out, it could be too late. So I think there's a lot of hope and possibility for improvement down the field. Yeah, it's really important work.
Yeah, absolutely.
So how does alcohol affect a developing brain? Dig into the science a little.
So for the craniofacial birth defects that we see with fetal alcohol syndrome, we know that the effects of alcohol are primarily due to inhibition of the sonic hedgehog pathway. Sonic hedgehog is a morphogen that is really important during gastrulation and neurulation, two really important events in developing the central nervous system and the face that develops around the brain. And we know that if you inhibit the sonic hedgehog pathway, you get a whole spectrum of birth defects called holoprosencephaly, which is a really severe defect of the brain and face. It was classically defined by cyclopia, which is its really in its most severe form. And we know that alcohol, when the embryo is exposed during these really early embryonic events, that alcohol inhibits the sonic hedgehog pathway, and that's one mechanism through which alcohol has an effect.
The sonic hedgehog, again, the name always distracts, I think.
It's a good name.
Yeah, whoever named it had found a lot of fun with that. Not just the speedy blue hedgehog character that you might be familiar with. It's essentially a master regulator of cell proliferation and development.
Absolutely. It is essentially a growth factor. Okay. It is important for development of the early neural tube. When you don't have sonic hedgehog, your early neural tube doesn't grow.
Mhm.
And even small inhibitions of sonic hedgehog can have really long-lasting effects, because building that early neural tube is what leads to our brains.
And is that during the 3 to 4 weeks that
Yes, so
sensitive?
the gastrulation, which is when we get to the 3 layers that will make up our entire body plan, happens during the end of the 3rd week, and that lays down the plan for developing the neural tube, which begins about the 4th week of development.
Yeah, I'm gonna make sure to toss in the show notes some like visual aids, because it's really hard to for this over a podcast. If you're looking at the video, it's kind of like there's a ball of cells, and then it kind of winds in on itself, if I remember correctly.
Right.
I'm doing a terrible job. But
No, so
I'll put a video
it's not an easy thing to grasp, especially without visual aids.
Yeah.
The gastrulation is essentially when we go from a flat sheet of cells to the 3 layers, the endoderm, the mesoderm, and the ectoderm, that will go on to form our entire body. And then it is that ectoderm that will go on to form our central nervous system, as well as our skin that overlies everything. And interestingly enough, while all of our muscles and bones and connective tissue are formed from that middle layer, the mesoderm, our face and skull are actually formed from the ectoderm, the same tissue that forms the central nervous system. And that's one of the reasons why during these early developmental periods, the face and the brain are developing together, because both the face, which is developed from neural crest cells, and these neural crest cells are derived from that ectoderm that will go on to form the neural tube. And so when we inhibit things like sonic hedgehog, then we're affecting both the brain and the face that is developing around it, because they develop from the same basic embryonic tissues.
Yeah, that's fascinating. I had forgotten this from my developmental biology classes long ago. The face and the central nervous system are part of that single pathway, that single sheet, right? And so sonic hedgehog is more effective or more required for the ectoderm.
It is absolutely
than the other layers, right?
It is absolutely required for the ectoderm. In fact, sonic hedgehog is, as the neural tube develops from a flat sheet of cells into the round tube, hence the neural tube, sonic hedgehog is expressed on that ventral surface of the neural tube and helps it to grow and helps those cells to proliferate. And
the inside layer, sonic hedgehog is
doing
all of its work. Yeah,
exactly. And there are many other morphogens that are expressed in other parts of the neural tube. We just happen to know that alcohol during the period of gastrulation and neurulation affects sonic hedgehog. And as an aside, alcohol is a really unique drug in that it affects different sets of cells at different periods of development. So during neurulation, for example, it can affect the sonic hedgehog pathway in the ventral parts of the neural tube. During later parts of development, it will affect dorsal parts of the neural tube. So different patterns of alcohol exposure typically lead to different patterns of brain malformation.
Mm-hmm. That's also the reason why there's like no safe time and no safe dose.
Exactly. It's just because we have the really severe brain defects that are similar to holoprosencephaly during the periods of gastrulation and early neurulation. Later patterns of alcohol exposure also cause defects that will affect the cerebellum, for example, or other parts of the cortex.
Yeah, that's fascinating. It's kind of scary knowing just how sensitive the embryo is. It's really not that many cells beginning. When you think about what eventually comes out as a full fetus.
It's really not. In the periods that we're talking about, the entire embryo could fit in the zero on a penny. It's so small. It's less than a millimeter.
Mm.
But at the same time, there are so many important embryonic events happening at this time that even small perturbations in development can really lead to long-lasting effects.
Kind of knock it off course.
Exactly.
Yeah, it's really scary, and I think people are becoming a little more aware of this, but knowing the science behind it and just how sensitive an embryo age, people understand how imperative it might be. And again, not to shame people. This is an eye opener for myself as well, right? Just a reminder that I need to think about this more.
Yeah, it's. I think it's something that we as a society need to think about, and we know that that even for adults, alcohol affects us negatively,
Right.
And it's again something that we just need to be aware of and try to do in moderation, possible.
Yeah, I've really cut down. There's even in the adult brain, there's all these studies coming out how there is essentially no safe dose for adults either, and people will say this is conflicting data and stuff, but the more rigorous studies are coming out and saying, yeah, it's not as essentially safe as we thought it was. Again, I'm not trying to shame people. I know that sometimes the stress relief maybe outweighs the benefits, and that's an entirely different discussion.
I don't get into what alcohol does during adulthood. I stick to early development, but it the science is still ongoing as far as adulthood. I think there's nothing wrong with a glass of wine or a beer, and even occasionally more, but we just need to be aware of what we're doing.
Yeah.
And I think that that's particularly true for someone who is pregnant. We don't know what the safe limit is for a developing embryo or fetus. And one of the reasons we don't know is because of those genetic factors. We don't know what those genetic factors are, and what might be safe for one person may not be safe for another.
Yeah, pregnancy can really be a minefield in terms of what you can and can't consume.
Absolutely.
I mean, there's foods and there's a million things that exposure, atmospheric exposure, and pollutants. We go on and on. So I mean, it is just so tangible thinking. And again, I will toss a visual aid up for people who've never seen a developing embryo. It's really not that big. It's tiny, and it's this little blob of cells, and dumping anything on it, essentially exposing it to anything that could be harmful. Gonna be scary.
Right, and people, as we become more educated, people have gotten really good about avoiding some of these things that we know to be dangerous. And part of my job is helping people to figure out what is dangerous, and that's what led us to our cannabinoid work.
Right. So that was really what caught my eye. You published a paper recently. You looked at both ethanol exposure in the developing embryo and cannabinoids, which are present in marijuana. There's THC, and then cannabidiol, CBD, which is people have really been buzzing about lately. Seems to do great things, but maybe not in the embryo.
Right. There is a lot of evidence that THC and CBD may have some beneficial effects. For example, cannabidiol or CBD is approved for children with intractable epilepsy. So epilepsy that nothing else seems to help. In some of these kids, it seems that CBD might help. And so it really does have its benefits. But as my lab has been showing recently, that again, during these very early developmental periods, both THC and CBD seem to be having the same effect on the brain and face that alcohol does. And what we've also gone on to show is that these effects of CBD and THC seem to be greatly exacerbated when combined with alcohol. so that we actually get a synergistic effect when we combine the two drugs, even at fairly low doses.
Oh.
So we get a synergistic effect of these drugs.
Yeah, to touch on that a bit, there's a concept in pharmacology called dose dependence, and we mentioned it a little before. The more alcohol you ingest, the more it'll affect the embryo.
Right.
I presume the same with THC or CBD. When you're combining them, the synergistic effect, it's almost like even at both are at low doses, they can amplify each other.
Yes, because what we went on to find was that both CBD and THC are also affecting the sonic hedgehog pathway, and so both CBD and THC can inhibit smoothened, which is a really important G protein coupled receptor that helps amplify the sonic hedgehog pathway. And so inhibition of that one protein can really shut down the sonic hedgehog pathway.
So the neural tube doesn't develop properly, and everything we talked about comes to fruition. So that's really scary because they are commonly used together, alcohol and marijuana. And I get this sense that, while marijuana toxicity isn't exactly the same as ethanol, people are kind of thinking of CBD as this wonder drug, and it absolutely is really helping kids, right? The issue I've seen is that people often refer to it as completely innocuous, and your research suggests that it's definitely not the case.
Yeah, people tend to think of CBD as being innocuous because it's not a true CB1 receptor agonist. So going back to the pharmacology, THC and other synthetic cannabinoids, they are true CB1 receptor agonists, and CBD is not. But what we're seeing here is that CBD is affecting the sonic hedgehog pathway by inhibiting smoothened, thus downregulating sonic hedgehog pathway activity. What is making this matter worse, I think, is that we know in the mouse that THC is worse than CBD. So even taking equal doses, THC will have more of an effect than cannabidiol. But what we found, and the reason we think for that, is that in the embryo, the CB1 receptor is dimerizing with smoothened. And so in the embryo, during normal sonic hedgehog pathway activation, smoothened activates the downstream pathway, and you get normal cell proliferation. Without any cannabinoids on board, the CB1 receptor is dimerizing with smoothened and helping to prevent during normal development, the CB1 receptor seems to be dimerizing to smoothened and helping to fine-tune the sonic hedgehog pathway activity so that we get a very normal development. However, when you expose the embryo to something like THC, it's activating the CB1 receptor, which inhibits smoothened, but THC also seems to be directly inhibiting smoothened as well. So you're getting inhibition of the sonic hedgehog pathway in two ways.
So they're physically, the CB1 and the smoothened receptor are physically connected.
As far as we can tell, they seem to be physically dimerizing.
Wow. And does that happen in adult brains, as far as you know?
It does. We have a little bit of evidence to show that it does. And for the CB1 receptor, this is not terribly unexpected. We know that the CB1 receptor, which is also a G protein coupled receptor, tends to dimerize with a lot of other G protein coupled receptors, and that's been shown in the adult brain literature quite a lot. So in retrospect, it really shouldn't surprise us that CB1 is binding to another G protein coupled receptor. It has just never been shown before, partly because smooth end and the sonic hedgehog pathway in general is far less important in adulthood than it is during early embryonic development.
Yeah, and so that, I mean, you've uncovered what seems to be a very clear link between how marijuana is affecting this development, and with this kind of CBD craze, just in general, marijuana legalization is becoming prevalent. It seems like every other month there's a state that's looking at legalizing it. A lot of people are jumping up for joy, and in many cases, in the adult brain, seems to be less dangerous than ethanol. But this work is so important. I think we may 40 years from now be looking at it the same way as we look at fetal alcohol syndrome.
It's very possible. I think we could very well be looking at a major public health crisis.
Right.
With, like I said, I think that there may be a lot of beneficial effects of the cannabinoids, but much like any other drug, we just have to be careful with how and when we use it.
And how we use it is also another thing that seems to really be cropping up lately. There's all of these different delivery mechanisms for cannabinoids, and there's like gels and put it in food now. There's the oils, and with this dose dependence in mind, essentially the more of that compound that you ingest, the more it could affect the embryo. These delivery mechanisms that allow you to essentially just dump these compounds in your system can become really, really dangerous, I think. And again, I'm not trying to scare people, but just be aware that this stuff is more potent than it ever was.
It really is. We in the mice we tested dosages of THC and CBD that are comparable to what people use on a regular basis, or some of the therapeutic doses of cannabidiol. But the scary part for me is that so much of particularly the CBD oil industry is unregulated, and we don't know how much CBD is in these products.
Right.
So we really have a lot of work to do to sort of identify how much CBD it takes to impact development, because it might be just a little bit, but at the same time, if you're using a lot of CBD products, it would really be affecting early embryonic development. But it's hard to know how much CBD is in these products and how much you're ingesting. It's not like alcohol, where the percentage of alcohol is on every bottle,
well tested. And
exactly.
I mean, this comes with, I think, every new drug has to go through a process for the FDA. Essentially, like any drug that goes on pharmacy shelves has this whole process. Because we're going with this state-by-state legalization, it's this really unique scenario where it's kind of like small businesses are cropping up everywhere and selling their products, and this home craft beer brewing sort of scenario.
It is, and I think it's leading to a lot of different products across the country. I think if you looked at 20 or 30 years ago, nobody overdosed on marijuana. It just didn't happen. But that's because over the last 20 to 30 years, the concentration of THC in marijuana has increased, in some cases by 20-fold. And so that has led to numerous instances of overdoses from marijuana, which if you get too much THC, it can have some really scary effects. And it can cause psychoses. It can cause a lot of other physiological effects.
Yeah, and again, it's a scary time to be developing embryo, especially, right? There's all these compounds swimming around that. Again, it has a reputation of being harmless. In many cases, it's less dangerous than alcohol, at least epidemiologically, if you look at broad studies. But it certainly has effects, and I'm glad you're on the case, really.
It does, and it's interesting that you bring up epidemiology. We know from studies back in the 80s, for example, that kids with prenatal marijuana exposure had, issues in school. They had some minor learning disabilities. But really, with the more widespread use of marijuana and the increasing THC concentrations in marijuana, we really need to do all of these epidemiology studies again, because now we have a lot more kids potentially exposed, and the concentrations of these compounds are so much higher now than they were back in the 80s earlier.
Yeah, it's really sad that research has been blocked in this area for so long, and the doors on research seem to be opening just as the floodgates on all of these substances are opening too. So there is going to be this Wild West period where people are going to have access to all this and not know fully what they could be doing. So yeah,
it will be, and I think the illegal aspect of marijuana has also made studying it difficult, because nobody wanted to tell a researcher that they were using marijuana, simply because they didn't want to get in trouble for it.
Or their doctors, even.
Or
worry
their doctors.
about that, yeah.
Yes, and there are some places where if you will get locked up if you're on drugs, and so that really has impeded our epidemiology research. So, but going forward, hopefully we'll do a better job at that, getting at those questions.
Yeah, I think people are becoming a lot more open to it in general. Comes with, again, some of the things we talked about, the bad side, but there's also good news in that we'll understand.
Hopefully.
Along that note, is there anything you're really excited about? It may not be your research, but I'd love to know anything in your lab that you're able to share, of course, because publication barriers right now.
I think that one of the more exciting areas of interest of research that we're getting into is again looking at mechanisms. We're starting to better understand some of the mechanisms through which alcohol works, as well as these cannabinoids. For example, sonic hedgehog signals through these little cellular antennae called primary cilia, and these primary cilia are absolutely critical for development. And in fact, if you have a mutation in a primary cilia gene, you can develop what is called a ciliopathy. And some new research from our lab is showing that one of the ways through which alcohol might be working is by affecting these primary cilia, which are really just little extensions. They're
of a single cell, right?
They're right. They're like little antennae hanging off of a cell, and almost every cell has one. And they basically work by taking out or extracellular signals and transducing them into the cell. And for example, sonic hedgehog is one molecule that works through primary cilia, and we're starting to see that alcohol might be affecting primary cilia.
So as a biologist, I always look for specificity. So it's how narrow can you find your window, and then you can extend that to pharmacology, where you're looking for a therapeutic target. In some cases, many of the drugs we use in like cancers, for example, aren't very specific, so hit a lot of pieces of the cell. But in this case, if things are happening through the primary cilia, and that's the mechanism of alcohol inducing cells, then maybe we are on the road to a therapeutic target that wouldn't affect the other functions.
Right.
I know this is very preliminary.
It is. And, one of the interesting things about a lot of what we do, even though we focus on early embryonic development, what other researchers have learned over the years is that these primary cilia play a key role in a lot of cancers. But at the same time, the sonic hedgehog pathway also plays a role in a lot of cancers. And there is some interesting new work looking at perhaps some of these cannabinoids may play a role in help as a chemotherapy agent. And so it's interesting that our work during early embryonic development is actually coinciding with a lot of cancer work, looking at these basic cellular processes that may play a really important role in embryonic development as well as cancer. So it's an interesting overlap.
Yeah, to someone uninitiated, it seems really counterintuitive, but cancer is a disease in which you have this rapid proliferation and growth of cells, and so in an embryo, that's exactly what you want.
That's exactly what's happening.
Yeah, but in cancer, you're already developed. You don't want, parts of your liver to suddenly start developing rapidly and doing their own thing.
Exactly, and in cancer, it's I'm not a cancer researcher, but it is an uninhibited and uncontrolled growth. And in some respects, embryonic development is a really rapid period of proliferation. But at the same time, it's also a very fine-tuned proliferation, and that everything is happening in the embryo in a very precise spatiotemporal pattern in order to develop into us.
Yeah, it's really fast, but really well organized.
It is very well organized.
Yeah, truly amazing, which makes sense as to why if you throw just about anything into any tiny wrench into the machine, things can go wrong so quickly.
Exactly, and that's why the developing embryo has so many processes to control exactly what is happening and what cells are proliferating where and what cells are migrating where, because we really do need to take essentially a ball of cells and turn them into us.
Yeah.
Some of the most complex organisms in the universe.
Over just 9 months. Yeah.
Right. Or 25 years, depending on.
That's true, yeah.
Some of us are still developing.
And there's some evidence that, at least some parts of your brain are still moving things around after 25, which is good news for me. I'm 27 now. So when I hit 25, I thought something would magically change. Dr. Parnell, I really appreciate it. This work is going to get more and more important. I think getting the word out, especially, again, we have known about the effects of alcohol for a while, but marijuana is rearing its head, and there are going to be some issues that I can see down the road.
I think so. We're really in the beginning stages of this work. We have a long ways to go. We have a long ways to understand exactly what is happening. And we have a long ways to go to understanding the epidemiology. But I think that people would be wise about being moderate and being careful, particularly if they're trying to get pregnant or are pregnant.
Yeah.
And until we know more about it, it's always better to be on the safe side.
Safe than sorry,
Exactly.
absolutely. Can't agree more. A little bit about you, I think.
Yeah, no, that's fine. So I have studied fetal alcohol syndrome my entire adult life. I actually started as a freshman in college working in a lab that studied the effects of alcohol during the 3rd trimester. And there are some very unique effects that alcohol has on the brain during the 3rd trimester. And I then stayed in that lab for grad school. This was at Texas A&M University in the School of Medicine. And so I did my PhD in that lab. I started out working with rats. And by the end of grad school, I was actually working with sheep. Yeah, so rats and mice are interesting in that their equivalent 3rd trimester in terms of their brain development is actually postnatal. So mice and rats are born far more premature than humans are, particularly in terms of brain development. And so in order to study 3rd trimester development, you actually have to study them in after they're born. sheep have the advantage of they're actually more mature when born than we are. So their third trimester equivalent in terms of brain growth is actually completely in utero. So if you want to study maternal factors that may play a role during third trimester development, it's better to do it in an animal model where where you actually have a maternal factor.
Yeah, that's really interesting. I didn't think about that because in your previous paper you do use mice, but you're looking such an early stage that everything is practically the same.
Exactly.
So, yeah. For reference, in the development, you can kind of think about it like sheep come out almost ready to go and I can walk around. Human babies cannot, certainly.
Right.
And then mice come out, basically they can't really fend to themselves.
Right. Mice, they can crawl around, but that's about it. So and it's simply a function of brain maturation. So in mice we completely study them in utero when in terms of first trimester equivalent, because the great thing about using mice or rats is that the basic developmental processes that occur during early development are almost identical across all vertebrates. So we can actually see a lot of these same effects of alcohol in zebrafish, right, for example. So zebrafish make a great model. In fact, a lot of our work with alcohol and cannabinoids was done with collaborators who use zebrafish. But
basically just little minnows for having worked with that.
They are. They're cute little minnows. My labs exclusively studies mice, but I collaborate with a lot of people who use zebrafish, and they're great models because you can do a lot of genetic manipulations. You can do a lot of fine-tuned drug exposure and they're developing just within their own little eggs. Well, they're developing outside of a mother, so you can watch them, you can see them.
They can be transparent.
And they're transparent, so you can really see what's going on. But after grad school, I actually came here to UNC to work in another fetal alcohol syndrome lab. And this time, this lab was working on very early embryonic development. So I did my postdoc here at UNC, and I stayed here as a faculty member. It is a great spot. I love being here at UNC.
Outside of the lab, do you have any hobbies that? No, you're pretty busy as a faculty member here at UNC.
No, but I still find time to spend time with my family. I have 2 kids. Coach my son's baseball team, and we have a lot of fun. Traveling whenever we can. Both kids do a lot of sports, so there's a lot of sporting events and other typical kid events that occur.
A lot of places to be. Yeah,
exactly.
I'm sure that keeps you busy. Yeah, I think it's I preach about this probably too much, but it is important to keep it balanced. We want to be in lab as much as possible. This work you're clearly like very fascinated with, been following since you were an undergrad, so I know you're committed to it. But it gives you that perspective.
It is. It's super important to maintain a nice, healthy balance. If you are in the lab 24 hours a day, and if you like that, that's fine. But if you do have outside activities, it's good to provide balance to both of them. It's especially true when you have a family. You got to make time for them.
Yeah, absolutely. So, yeah, again, Dr. Parnell, I really appreciate it. Are there any final thoughts for the audience? Could be advice for people trying to get into the field, or kind of anyone casually listening?
I think the most important thing for any potential scientist or anybody looking at trying to determine what area that interests them or what field to go into, I think that Oliver Smithies, who was a Nobel laureate here at UNC, as he once said, that you have to be passionate about what you're doing. If you don't love your science, don't do it. Mm-hmm. Don't go find something else that you love, because if you don't love what you're doing, you're not going to do it very well, or at the very least, you're going to be miserable. Yeah. Don't be miserable. Go find something better to do. Go find an interest that really interests you. Mm. And that you're passionate about, and then you can really throw yourself into.
I think it can be hard to know for people who are just getting into the field. They might find something moderately interesting and they think that's it, but I don't know. How did you know, I guess, when you were working in the...
I think it's evolved over the years. Mm. Many times, especially as a grad student or even as a postdoc, you're sort of given a project and you may or may not like it. But I think the thing to do is to work hard to evolve that project into something that you love. Mm. I'm lucky now as a faculty member. I get to choose what I work on, and that's part of the great thing about being in academia is that you choose what you work on, and will hopefully choose something that you love. I think for young scientists, it's important to really think hard about what you like and what interests you and what you don't like, and choose, for example, choose your rotations wisely so that you pick a rotation that you think you'll love, and you'll find out soon enough, hopefully, whether you do or not.
Yeah.
But
it wasn't like love at first paper.
It wasn't. I actually for many years, I actually thought that I wanted to go into receptor pharmacology.
Doing a little bit of that now. I
guess. It actually turns out that I went away from it for many years and looked at more cellular or looked at larger global behavioral differences, for example, or but yes, I actually have kind of come full circle in that I'm now doing a lot of, I won't say I'm a pharmacologist by any means, but we are looking at a lot of different receptors and how they may impact development.
I saw the dose response curves in in the scientific reports paper, which of course I will link in the show notes. So, but yeah, I mean, I didn't have the love at first paper experience myself, so just I think that's good advice for people, is that it will kind of grow into itself if you're on the right path. It may not feel immediately like the thing.
It might not, and science is often tedious.
Yep.
You have to work through it, but it often pays off in the end. Especially again, if you're doing something that you love, or you're working on a project or a problem that is really important. Mm-hmm. It's important. It's also important to keep the overall picture in mind. It's knowing that you're working on a project that will improve humanity is important as well.
Why we do it, at least most of us.
Hopefully.
Yeah, everyone I've met, I will say that.
We're not in it for the money.
Yeah, that's for sure. Yeah, thanks again, Dr. Parnell. I really, really appreciate it.
Absolutely.
Thanks for listening, all.